Hi there, I'm an IGM antibody, ready to show you how I function through both the processes of transcription and translation. First off, I should tell you a little bit more about myself. I'm officially named an IGM gene, more specifically immunoglobulin M. I work within the immune system, a system in which cells, tissues, and organs work together to defend the body from attacks from foreign invaders. I am found within blood and lymph fluids, my main job being to destroy pesky bacteria and infection. I am located on chromosome 14, specifically on Q32.33, and I have approximately 4,511 nucleotides. Right now, I am trapped inside the nucleus as a list of instructions. It's like a prison in here. I've heard the cytoplasm is absolutely chaotic though, but I want to see it for myself. But first, I have to form. The first step would be through the process of transcription, which changes my instructions in DNA into pre-mRNA. DNA contains my instructions within nitrogen bases such as A and T and C and G, and is a double-stranded helix. In the initiation stage of this process of transcription, RNA polymerase, along with the help of transcription factors, which are the green things on top, move down a single strand of DNA, reading 3 nucleotides at a time, such as TAC and then onto GCC, unwinding the DNA strand as it goes. As it unwinds, RNA is built using the DNA strand as a template, matching the nitrogen bases with complementary base pairings. Eventually, the RNA polymerase will meet a stop signal at the end of the DNA strand, which signals that it's time to stop copying. The newly made pre-mRNA goes off and the DNA strand comes back together. Now we finally have pre-mRNA. Our newly made pre-mRNA strand is almost identical to a single strand of DNA, except that T gets replaced with a U. My instructions that were once in that DNA strand are now finally in pre-mRNA, and now we are ready for the next step of processing mRNA. RNA processing makes changes and additions to pre-mRNA to ready itself for the next step of translation and before it can finally become a mature RNA strand. First, the intron is removed because this does not code for any of my IgM antibody information. The exons, which are the other letters, stay behind, and now we have this strand of RNA. Before it can finally become mature, however, we must add a cap and a tail. This ensures safety as it travels through the cytoplasm to my next location. We finally have mature RNA. In translation, RNA finally leaves the nucleus and enters the cytoplasm. Our RNA, a ribosome, attaches itself to 3 codons in the RNA strand, for example, CGG. A ribosome can only read 3 codons at a time as it codes the information. tRNA, which stands for transfer RNA, attaches itself to these 3 codons with anticodons, which match up with the codons as opposite pairs. tRNA transfers amino acids. Once tRNA is done translating the 3 codons, it moves on with a new amino acid. The amino acid stays behind and eventually connects itself with the other amino acids with peptide bonds to make a polypeptide. I'm finally one step closer to being able to leave the cytoplasm, but first my polypeptide, aka protein, must go through the important step of protein conformation. Right now, I am a simple shape, so I am a primary. In order to be my true self, which is a little bit more complex due to my order of amino acids, I must fold myself multiple times to finally become the conformation shape of quaternary. I'm finally an IgM antibody, but I'm still stuck inside the cytoplasm. I need to leave the plasma membrane of the cell wall. I must go through the process of exocytosis to do so. A vesicle is made from a nearby Golgi body, and I hitch a ride on top of it in order to leave the cell. The vesicle transports me to the plasma membrane of the cell wall, but I need to leave, so the 2 become 1, and then pop, I'm out, and the cell wall closes back out, and the vesicle goes away. And now I am free! As an IgM antibody, I go somewhere in the body where I'm needed, such as the bloodstream, to fight off bacteria and infection. Thanks for watching me on my protein synthesis journey.